Okay, chapter 8. So, in chapter 8, we have chapter 8.4, which is transcription. Now, before we learn about transcription, there's some background information that we have to know. So, there's DNA and RNA. DNA is called deoxyribonucleic acid, and RNA is ribonucleic acid. The only difference between these two is that DNA has this deoxy part, while RNA does not. So it's pretty easy to memorize. Now, as you can see here, DNA has four types of nitrogenous bases, and they are A, T, G, C, while RNA, the only one that's different is the U. So U is uracil. They have AUGC. And if you see here, A and T bond together with two hydrogen bonds, while G and C bond together with three hydrogen bonds. The same thing is for RNA, but except the T and the U are replaced. Now, if you see here, this this thing is DNA, while this is RNA. So essentially, RNA is like half of DNA. So DNA is something what we call double helix. So there's two kind of strands, while RNA only has one. Now, the process of the whole DNA thing is goes from replication to transcription to translation, and we're going to talk about this more later. Now, here is what a nitrogenous base looks. No, not a nitrogenous base. What a nucleotide looks like. Yes. So first, this hexagon, I think this thing here, pentagon here, is called pentose sugar. The name of it is pentose sugar, and on the left we have this phosphate group, and on the right we have the nitrogenous base. Now, the second is this thing called central dogma. So central dogma, essentially, it's the flow of genetic information from DNA to RNA to proteins. And so, in other words, it's like the relationship between the molecules and the processes. And the final product that comes out from central dogma is protein. And protein is what creates the whole living body. So there are three stages: replication, transcription, and translation. So replication is DNA copying itself to make more DNA. And so it's a process where there's a strand, it's pre-existing. So it means it was there before. And the before strand, it becomes a template in order to make a new strand of DNA. And what this is called a semi-conservative process, which means that one strand of the DNA, one is old while the other one is newly made. The second is called transcription. And what transcription is that it's the conversion of DNA message into an intermediate molecule. So it's DNA into making RNA. Transcription is literally just making RNA. And so the product of transcription is RNA, and RNA is then made from the DNA. Next, we have translation, which is when the RNA message is interpreted and it makes protein. So translation is the making of protein. And so the information that's inside the mRNA, which is messenger RNA, it kind of is translated into sequences of amino acids which are then combined to make protein. Now, the thing is, there are prokaryotic cells and eukaryotic cells. Prokaryotic cells are like single-celled organism, while eukaryotic cells are, like human beings. We have multiple cells, and we need multiple cells to live. Each cell has a different function. So in prokaryotic cells, all of these stages happen in the cytoplasm: replication, transcription, translation, all in the cytoplasm. Whereas for eukaryotic cells, only replication and transcription happens in the nucleus, while translation happens in the cytoplasm. So if you see this picture here, replication, the making of DNA, and transcription, the making of RNA, all happens inside this blue thing, which is the nucleus. And then later it goes out in order to do translation, in order to make protein. And this green space outside of the nucleus is called the cytoplasm.